Good morning, students. Today we are discussing about the topic is physiology of digestion. Previous class we are discussing about the physiology of digestion in the digestion of bowel cavity and the digestion of stomach. Today we are discussing about the topic is digestion in small intestine. The topic name is digestion in small intestine. Now, so small intestine. Previous class we are discussing about the associated glands of intestinal glands. So intestinal glands is secreting to the intestinal juice. Next, in this intestinal glands is consist of the two types of cells. First one is the Brunner's gland. First one is Brunner's gland. And the second one is crypts of Lieberkühn. The intestinal gland is consist of the two types of glands. First one is a Brunner's gland and the second one is a crypts of Lieberkühn. So in this Brunner's gland and crypts of Lieberkühn, both are secreting to the intestinal juice. These glands are secreting to the intestinal juice. In this intestinal juice is consist of the peptidases. It is consist of the peptidases and disaccharides. It is consist of the peptidases and disaccharides. So first of all, today discussions about the topic is digestion in the small intestine. So intestinal glands. Intestinal glands is consist of the two types of glands. Brunner's gland and crypts of Lieberkühn. This kind of glands is secreting to the intestinal juice. In this intestinal juice is containing to the peptidases and disaccharides. So peptidases like tripeptidases, dipeptidases and aminopeptidases. At the same way, disaccharides like these are totally sucrose, maltose and lactose. These are the examples of the disaccharides. These are totally topics of intestinal glands. Now today discussions about the topic is digestion in small intestine. Now the digestion in the small intestine, in this small intestine is digestions of the first one is the digestion of proteins. The first one is digestion of proteins. First one is digestions of proteins. Now, in this digestions of the proteins, in this process, the intestinal glands is in the small intestine is containing to the inactive stage of the trypsinogen and chymotrypsinogen are present. Trypsinogen and chymotrypsinogen are present. Now, the digestion of the proteins in the small intestine. In this process, the small intestine is containing to the inactive stage. These are inactive stage of the trypsinogen and chymotrypsinogen are the present. Next, in this inactive stage of the trypsinogen and chymotrypsinogen is activated by to the enzymes is a enterokinase enzyme. This inactive stage of the trypsinogen and inactive stage of the chymotrypsinogen both are is activated by to the enzyme is a enterokinase. Enterokinase. It is activated to the inactive stage of the trypsinogen and the inactive stage of the chymotrypsinogen. Now the equation form is the trypsinogen, inactive stage of the trypsinogen. Inactive stage of the trypsinogen. It is a inactive stage. Inactive stage of the trypsinogen is activated by to the enterokinase enzyme. Is activated by to the enterokinase enzyme. Is activated by the enterokinase enzyme. Finally, the inactive stage of the trypsinogen is converted to the active stage of the trypsin. Is converted to the active stage of the trypsin. So now discussions about the topic is digestions of the proteins in small intestine. In small intestine is containing to the inactive stage of the trypsinogen and chymotrypsinogen. Trypsinogen and chymotrypsinogen. In this inactive stage of the trypsinogen and chymotrypsinogen, both are is activated to the enzyme is enterokinase enzyme. Finally, the inactive stage of the trypsinogen is converted into the active stage of the trypsin. At the same way, the chymotrypsinogen, it is the inactive stage of the chymotrypsinogen. The inactive stage of the chymotrypsinogen is also activated by the chymotrypsin. It is the active stage in the presence of trypsin. So it is the digestions of the proteins in small intestine. So the digestions of the proteins in the small intestine, so it is consist of the inactive stage of the trypsinogen and inactive stage of the chymotrypsinogen. Both are is a inactive stage. In this inactive stages, enzymes is activated to the enzyme is a enterokinase enzymes. So the trypsinogen, inactive stage of the trypsinogen is activated by to the trypsin in the presence of the enterokinase. At the same way, the chymotrypsinogen is inactivated and is converted to the activated of the chymotrypsin in the presence of the trypsin. Now we are observed in this equation, the in this same enzyme is used to the in this equation. This type of process is a autocatalysis process. This process name is autocatalysis process. So what is the meaning of autocatalysis? Autocatalysis means same enzymes used in the activations of the equation. This type of process is known as autocatalysis. It is a 2 marks question. What is autocatalyzations? So the autocatalysis means same enzyme used in this activation form. This kind of process is known as autocatalysis. It is the proenzymes is a inactive stage to active. Now the discussions about the digestions of the proteins. So in this digestions of the proteins, in this process, the chymes enter into the small intestine. In this process, the chymes enter into the small intestine. So when the chymes enter into the small intestine, the chymes consist of the proteins. The chymes consist of the proteins, peptones, and proteases. So the chymes enter into the small intestine. The chymes consist of the proteins, peptones, and proteases. Now, in this proteins, peptones, and proteases is convert into the tripeptidases and dipeptidases. Is convert into the tripeptidases and dipeptidases. So in this proteins, peptones, proteases is convert into the dipeptidases and dipeptidases. Tripeptidases and dipeptidases. In the presence of the enzymes is a trypsin and chymotrypsin. These are the active stage of the enzymes. How to form of the active stages? Because there is a present to the which enzyme? Enterokinase enzyme. In this enterokinase enzyme is a, so the inactive stage of the trypsinogen and chymotrypsinogen both are is formations to the activation form of trypsin and chymotrypsin. In this active form of the trypsin and chymotrypsin both are is activated to the proteins, peptones, and the proteases. Finally it is converted into the tripeptidases and the dipeptidases. After the tripeptidases, tripeptides is converted to the dipeptides and amino acids In the presence of tripeptidases. At the same way, the dipeptides is converted to the amino acids. Dipeptides. The dipeptides is converted to the amino acids. in the presence of the dipeptidases. Dipeptidases. Finally, we have formed to the amino acids. So the digestions of the proteins in this process in the small intestine is conditioned of the inactive stage of the trypsinogen and inactive stage of the chymotrypsinogen. Both are is an inactive stage. After in this inactive stages is formations to the active stage. In this using of the enzyme is a enterokinase enzyme. Finally, the trypsin and chymotrypsin is both are active stage. In this active stage of the trypsin and chymotrypsin is both are is act on to the proteins, peptones and proteases. Finally, they are formed to the tripeptidases and dipeptidases. And tripeptides is converted into the dipeptides and amino acids in the presence of tripeptidases. And the same way dipeptides is converted into the amino acids in the presence of dipeptidases. It is the digestions of the proteins in small intestine. Next, we are discussing about the digestions of the lipids. Continue to the next class.